the webinar.
Good morning, everybody. We're going to, as usual, wait a few minutes and then we'll get started. It's great to see you all. Look at all those people. Good morning, Thomas. Or good afternoon, as they would say where you are.
Yes, good afternoon, good morning.
And good night. That's like that Truman Show. I love that. Good morning, good afternoon, and good night. All right, we'll wait another minute or so before getting started.
Okay.
Just another minute. Thank you for your patience, everyone. Good morning, good afternoon, or wherever you are. Okay. So I think we'll get started. So welcome to this morning's lecture from the Physical Biology of the Cell 2020 edition. And it's truly my great pleasure to introduce today's speaker, Dan Needleman, who's a professor at Harvard University. So as usual, I'd like to start with some of the facts, and then after that, maybe turn to some interesting asides. So Dan grew up in Rhode Island, and I know that personally because his dad, Alan Needleman, was a professor at Brown University who hired me. And so we used to have these wonderful parties at their house and others, and I got to see Dan, the teenager. And it's really funny because not too long after that, Yane, who I see is here, he called me up one day while we were working on our book, actually. He said, yeah, there's this really great undergrad here who's majoring in physics at Brandeis, and that was our Dan. After that, he went to UCSB, where he got his PhD with Cyrus Safina, if I remember correctly, working, I think, on microtubules and maybe doing things related to X-rays and stuff, but I don't remember. After that, he went for a postdoc at Harvard University with Tim Mitchison, and at that point, I think began part of his long association with the MBL. I can't actually remember, Dan, but I suspect you were a student in physiology, and I almost have this weird idea it was the same year as Ernan, actually, which would have been maybe 2005.
No, I don't think
maybe.
so.
No, yeah,
okay. Yeah. Before that.
Okay. Anyway, so and then also he was a TA in physiology, and then in addition, Tim had a lab up there in Lerb on the 3rd floor, and Dan would spend whole summers there doing research. Do I remember that correctly?
That's right.
Working on Xenopus and stuff. After that, Dan became a professor at Harvard and has risen up through the ranks gloriously with a long series of really cool achievements. And what I would say is that, he's in this category of people, as is Joe Howard, who I see is now a panelist with us, and several others on this call, of the kind of people where you just don't know what the next thing's going to be. And that's really one of my favorite things to say in letters for tenure and prizes and this and that is, this is the kind of person that, when you go to meetings there's those people that you see and you are excited to see them, but you know that they're on a track and that they're going to tell you about their progression on that track. And then there's people like Dan, where you basically have no idea what the latest thing is going to be, but you know it's going to be done well. So today's talk, Biophysics of Mitochondrial Metabolism in Vivo, really took me by surprise, not in this context, because I'm now well aware of that going on in his group, but I have been deeply interested in the work that he's done, for example, with Jan Bruges on cytoskeletal proteins and spindles and so on. And I was really quite taken aback when I first heard about this stuff. Dan organized a really beautiful workshop that many of us on this call were at last December at KITP in the great days before the pandemic. I'm pretty sure we're all yearning for that. I'm actually in Santa Barbara right now, and so just came back from the beach, and it would be nice if everyone were here or at MBL. Anyway, so Dan's really a creative force and really knows how to make a measurement and knows how to think about those measurements in deep and very, very interesting ways. And this topic in general, how energy is used in biological systems to beat back the sad tempo of time of equilibrium, the pull to equilibrium, is one of the great topics. So anyway, it's great. Thank you so much. Dan comes every year to talk in our course, and he's one of the few repeat performers, and you're about to see why. Okay.
Thank you so much for the kind words and for the intro. It's always fantastic to be involved with this. And I wish I could also be on the beach swimming, but in future years. Okay, so like I was saying, I'm going to tell you about work we're doing on biophysics and mitochondrial metabolism in vivo. So my overriding interest, scientific interest, is in biological self-organization. I want to have a quantitative understanding of biological processes, of how molecules self-organize into subcellular structures in cells, and how cells self-organize into tissues and organisms, and how these are perturbed in disease and change over evolution. So that's the big picture. So in this talk, after giving the introduction, I am going to try to touch on 3 different topics, which we may or may not get to. All right, starting from the beginning. All right, so most of my work has actually been on cell division. So in cell division, the cell physically divides in 2, and chromosomes are segregated into the daughter cells. And so the part that my lab has mostly worked on is the structure, the spindle, which is this dynamic protein assembly, which is at the heart of cell division. So the spindle assembles and aligns chromosomes, and then it segregates chromosomes into the 2 daughter cells, and then it disassembles. All right, so my group has done a lot of work on different biophysical studies of the spindle, including looking at spindle assembly, positioning, and chromosome segregation using different types of techniques, different quantitative techniques, and with the goal of trying to gain insight by combining these quantitative measurements with some type of theory. And so that's mostly what my group has done. Lately, we've been changing and well, continuing to work on this, but also focusing on other directions as well, which is mostly what this talk is going to be about, these other directions. So chromosome segregation is important, and doing it right is important, because when it doesn't go right, things are bad, right? So if all was well, when you segregate chromosomes, half the chromosomes would go in one daughter and half would go in the other. Sometimes that doesn't happen. Sometimes you have errors. So you can get an extra chromosome in one of the daughters and lose a chromosome in the other daughter. And so this type of chromosome segregation errors are responsible or associated with cancer, infertility, and all other sorts of problems. So it's very bad when you have chromosome segregation errors. In particular, most spontaneous abortions are caused by chromosome segregation errors. Actually, age-related infertility in women is predominantly due to chromosome segregation errors in eggs and embryos. So if you look at if you look at the percentage of embryos with chromosome segregation errors as a function of the age of the mother, you can see that it increases greatly as the mother's age increases. So the primary treatment of infertility in people is in vitro fertilization. So actually 15% of all couples struggle with infertility, and roughly 2% of births in the US are due to a result of IVF treatment. And in other countries, it's even larger. So in IVF, what happens is that a patient comes in, that patient is given hormones that cause them to produce many eggs. These eggs are retrieved and mixed with sperm in a dish, so they're fertilized. And then those eggs eggs become embryos, and they're propagated as embryos in vitro, and then transferred back to the patient with the hope of having a child. So as I said before, age-related infertility in women is caused by increasing chromosome segregation errors in eggs and embryos, and these errors are a result of problems in the egg itself. And the way we know that to be true is as follows, is that if you look at IVF success rate as a function of the patient's eggs, that declines rapidly with eggs if a patient uses her own eggs. However, if a patient uses donor eggs from a young donor, then the success rate is essentially independent of the patient's age. And so really what's going wrong, what's causing this decline is going wrong in the eggs of the patient. Okay, so success rates of IVF are low. Only about 30% of IVF cycles result in a live birth. And the primary clinical challenge, what people believe is to actually choose like the good eggs and embryos to transfer back to the patient. And right now, the current way that's done is by looking at morphology. And so the clinicians or a embryologist will examine the developing embryo when it's in the dish and look at which ones are good, and which one looks good, and which one looks bad, and they'll choose the good-looking ones to transfer back to the patient. But obviously that's not as successful as one would like, which is why the IVF success rates on the whole aren't as high as one would like. All right, so we actually don't ultimately understand what goes wrong with eggs that leads to these errors in chromosome segregation. But a lot of people in reproductive biology believe that problems with mitochondrial metabolism may be one of the causes. Again, it's unclear if that's true, but that's a kind of a hypothesis in the field. Okay, so then now I'm just going to talk a little bit about mitochondria and the bioenergetics of mouse and human eggs. All right, so you have an egg sitting here. All right, so most energy supplied, is supplied by a breakdown of pyruvate. That's how eggs get most of their energy. And that breakdown, this oxidation, is carried out by mitochondria in the egg. And so mitochondria use this kind of breakdown to convert ADP to ATP. So they're the powerhouse of the cell, as we all know. And then cellular processes, convert ATP to ADP. And so you have this loop being driven by mitochondria oxidizing pyruvate. So that's kind of the overview of bioenergetics of eggs. All right. So then this kind of stuff has got me interested in the following question. What is, or is there a link between spindle behaviors and mitochondrial metabolism? So on one hand, we want to know what's the impact of mitochondria on the spindle? Do defects in mitochondrial metabolism actually cause defects in the spindle self-organization behaviors and chromosome segregation errors? We also want to know what's the impact of the spindle on mitochondria, and in other words, how much energy is used by spindle self-organization of behaviors. And from an applied perspective, we're interested in understanding if one can use a measure of mitochondrial metabolism to help select high quality eggs and embryos and hence improve IVF treatment. Okay, and one hint that, that this idea of a connection between mitochondrial metabolism and the spindle isn't, ridiculous is the following, is that perturbing mitochondrial metabolism does have an impact on the spindle. So people have shown that in different ways. Here's an experiment which Jingbo and Cullum, two postdocs in my lab, did. So mitochondrial metabolism requires oxygen to work. And so what they did is that they took mouse oocytes and they decreased oxygen levels, and you can see the spindle disassembles, and when they raise oxygen levels again, it reassembles. So decreasing oxygen causes a reversible disassembly of the spindle. All right, so the mechanism of this is unclear, and at least my yes.
You're probably going to say, and you've told me before, but I forgot. What's the fractional change in the amount of oxygen? And then there's also a question from Sri, which is there a good biological marker of age in this context? If you didn't happen to know how old the mother was, could you guess by simply examining the eggs? So that's kind of going backwards, and I apologize for not catching it before.
Yeah,
so
yeah.
Okay. Take hers first.
Okay. And the age one. So it's definitely true. People have shown that in eggs from older individuals, mitochondria are screwed up. Actually, that's true for most random cells in the body, that mitochondria in older individuals are screwed up. And people have also shown that there's a correlation between eggs which have screwed up mitochondria and whether those eggs actually produce a viable offspring. Whether that's a causative connection or not isn't clear, but there's a correlation there. In this particular experiment, if that answers Sri's question, in this particular experiment, the oxygen was reduced by a factor of over a factor of 100, and so this was very, very low levels of oxygen.
Okay, yeah, that's substantial.
Yeah. But like I said, so in this experiment, the mechanism isn't clear. You could imagine that has to do with ATP production, but it could also have to do with other things. Because so here I'm emphasizing the role of mitochondria with ATP production, but of course mitochondria have many other functions they carry out. And so then I would argue, at least for me, the way I want to understand this is to try to have a quantitative understanding of mitochondrial metabolism and a quantitative understanding of cytoskeletal and spindle bioenergetics, and then see how they're connected. Okay, all right. So that's kind of that's some of the motivation of trying to develop a biophysical understanding of mitochondrial metabolism in vivo. And so then I would argue that to obtain a quantitative understanding of mitochondrial metabolism requires a way to quantitatively measure mitochondrial metabolism and requires some quantitative theory of mitochondrial metabolism. So, like in this talk, I'm going to more focus on the first part, and then touch a little bit on the second as well. Okay, and that brings us to the first topic here, which is trying to measure and model mitochondrial metabolism in vivo. Okay, so there are many different ways of probing mitochondrial metabolism. One very powerful way is related to doing metabolic imaging of NADH and FAD. So NADH and FAD are two small molecules which are central to mitochondrial metabolism. And so they're used in nearly all ATP production that mitochondria will do. And it ends up that NADH and FAD are autofluorescent molecules. That's just how it is. And so people for a long time have been studying mitochondrial metabolism by looking at the fluorescence of NADH and FAD. However, just looking at the fluorescence intensity of those molecules alone is really not sufficient to quantify metabolism. There's another technique, which again, many people have done, which isn't just looking at the intensity, but using an approach called fluorescent lifetime imaging microscopy, or FLIM. And so there are many different ways to do FLIM. The way that I want to talk about is the way that my lab does it, which is called time correlated single photon counting. And the basic way this technique works, so it's not entirely true what I'm about to say, but we're just going to pretend it's true that it works this way, is as follows. Is that, okay, so you have a pulsed laser. Okay, so a pulse comes in your microscope, hits your sample, hits your fluorophore, in this case NADH, gets in an excited state, relaxes, and emits a photon, which then you detect. And you have very fast electronics, so you can detect the time between that pulse and the photon that you detect. So pulse one comes in, and then 3.78 nanoseconds later, you detect a photon. Next pulse, nothing. Next pulse, nothing. Next pulse, and 1.87 nanoseconds later, you detect a photon, etc. If you do this with a 80 megahertz laser, that's 80 million pulses a second, you can rapidly build up a histogram of photon arrival times. And FLIM, it gives you a histogram at every single pixel in your image. And so while, a normal image, like you would have a fluorescent intensity at every location, the number of photons, with FLIM, you don't just have the number of photons, but you have the full histogram of photon arrival times at every single pixel. Okay, and what people have known for a long time is that this is powerful for metabolic imaging because the lifetime of NADH depends on whether it's free or whether it's bound to enzymes. And so, for example, if you look at NADH in a mouse oocyte, so this is a metabolic imaging of NADH in a mouse oocyte, and you group all the photons together that you detect from this oocyte, then you can get some pretty histogram, which then and then you have this fast decay to the free NADH and the slow decay to the enzyme-bound NADH. And if you fit histograms like this using a model where you have kind of two exponential decay process, you can actually measure by looking at the relative amplitudes of these guys of the fraction of NADH bound to enzymes. And then by looking at the intensity, you can measure the concentration of NADH, basically. And so you can measure how much NADH you have and how much is engaged with enzymes. And furthermore, NADH is highly enriched in mitochondria, so the bright regions in this image are mitochondria. So you can segment out those images, in this case using kind of machine learning-based approaches, in which case you can specifically look at, in mitochondria, the concentration of NADH and the fraction bound to enzymes. All right, and so from what I'm going to talk about from now on, that's what we're talking about. FLIM of NADH in mitochondria in mouse oocytes. All right, so then one can do quantitative perturbations of mitochondrial metabolism and just kind of see what happens. So like we were talking about before, oxygen is required to run the electron transport chain, for mitochondrial metabolism. So you can just drop oxygen and see what happens to your FLIM measurements. All right, and so you take oxygen and you go from 5%, which is kind of physiological, and you drop it to as low as you can go. And then if you take a movie, what you see is that the NADH gets bright, and then if you restore it, it goes back.
Hey, Dan.
Yes.
One question that we have is, how specific is this approach to NADH versus other autofluorescent molecules that might be in the mitochondria?
Yes, yeah, and so here you can very specifically look at NADH and NADPH have very similar fluorescent properties, so you can't distinguish those two with this technique. But the fluorescence in this case comes, but the fluorescence you're measuring is coming overwhelmingly, if you use the right filters and stuff like that, from those molecules. It ends up that in mouse oocytes, NADH is people have measured this by mass spec, is you have 40 times more NADH than NADPH. So in this case, we can safely assume that all of the signal really is coming from NADH. And then based on, like how the fluorescence behaves, it's kind of consistent with that. But people have,
But there are no other small molecules that have like such a fluorescence lifetime profile floating around in the cell.
And so, I think, it's, so like, I think one can argue that like the signal here must be at least, like 80% coming from NADH. Okay. And so there could be other things as well, but it's really overwhelmingly from NADH.
Perfect. Good. Thank you.
Sure. Okay, and so here, from what I'm saying now on, we're just going to kind of assume it's all from NADH, and we'll see if that assumption kind of like bears out. But from other experiments, like I said, people, it's overwhelmingly coming from NADH. Okay, so now because we dropped oxygen relatively slowly, we can assume, and in this case, it's a safe assumption, that the mitochondria are behaving quasi-statically, and so we can just plot FLIM parameters such as NADH intensity and NADH bound ratio as the function of oxygen. of experiments. And you can see that as you decrease oxygen, NADH increases, bound ratio, the ratio of bound to unbound NADH decreases. Okay, so but then how do we interpret this? And so then again, I would argue that to really interpret this, you need some model of mitochondrial metabolism. Okay, so there have been many previous mathematical models of mitochondrial metabolism. Most of them are quite complicated. And many of them try to, for example, comprehensively model all the biochemical processes that people know to be there. The difficulty of that is that involves many equations with many unknown parameters, and it's very difficult to test such models or relate them to experimental measurements, such as this FLIM experiment I was just telling you about. So the question is, can we develop a predictive model, a predictive biophysical model of mitochondrial metabolism that allows us to really interpret those type of FLIM measurements? All right, and so here we want a more coarse-grained model, but one that's still based on the actual thermodynamics and chemical stoichiometry of what's known to be happening in mitochondria. Okay, so kind of what I showed you before was previously, like that mitochondria are oxidating pyruvate and hence, converting ADP to ATP. In order to, look at this, we have to open this up a little bit more. And so what's going on in a little bit more detail, obviously still highly simplified, is that pyruvate comes into mitochondria and is broken down. And what that does is that process, which is the Krebs cycle, converts NAD plus to NADH, and then oxygen, like which is required for the electron transport chain, can convert NADH to NAD plus, and so you have this kind of wheel happening here. Spinning this wheel, when you do this conversion, that pumps ions across the membrane of mitochondria from the inside to the outside. Those ions can relax due to just kind of leak, so-called leak holes in the membrane, or by ATP synthase that can take that can also relax this gradient, which relaxing that gradient is used to convert ADP to ATP. Okay, so now we're slightly kind of opening up what's happening in mitochondria.
Hey, Dan,
yes.
There's a background question that I think is worth attending to now. So the question was, why are we connecting drop of O2 and dysfunctional eggs to mitochondrial requirement of O2? Can't there be some unknown direct way of how O2 affects the spindle? So yeah, I see where the person's coming from. In other words, I guess they're trying to understand the logical connection or why you hypothesized that connection.
Well, okay, so like I was saying before, so there's a lot of reproductive biology work which argues that there's a correlation between having screwed up mitochondria and having chromosome segregation errors and misfunctioning spindles, and so yeah, so that's kind of an idea that's out there in the literature based on a lot of correlative evidence. So here now what we're trying to do is just try to have a quantitative understanding of mitochondria. And then we're going to try to use that quantitative understanding of mitochondria to eventually to help us ask this question about, is there a causative connection between mitochondria dysfunction in the spindle? And so here now we're just using oxygen as a way to perturb mitochondria. That's just the goal here.
Great, thank you. That was helpful. That was helpful. Thanks.
Yeah. Okay, cool. And so all right. Okay, so now let's kind of consider this a little bit more. And now here, what I'm talking about, I should have said this before. This is really work by Jingbo, who's a postdoc in the lab, who did the relevant experiments and all the theory, which I'll be telling you about. And so what Jingbo did is he said, okay, well, let's think about this one loop in a little bit more detail, this NADH loop, because after all, we're doing measurements in NADH and mitochondria. Let's think what's going on. And so then he proposed, let's have the following coarse-grained model. Let's just treat all of the electron transport chain as one kind of big effective oxidase, and let's treat all the Krebs cycle as one effective reductase of NAD plus. And so then we have this cycle here of NADH binding and unbinding to these two essentially enzymes in these two pathways. and then NADH being converted to NAD plus, which then can go back to NADH. And so in kind of physiologically, this loop would be driven in this direction. Oh, sorry. Okay, and so then one can have rates for each of these steps, and for everything I'm going to tell you about, these rates could be arbitrary, even nonlinear functions of everything, basically. And so yeah, we're not assuming any linearity or anything like that. There's all of these steps could be arbitrary nonlinear, based on arbitrary nonlinear functions. Nonetheless, we can still describe this loop this way. All right, so now let's think about this a little bit more. So let's think about this box here. We can think about the flux going through this path, this part of the pathway. And that will depend on the flux. We have a rate that NADH NADH bound to reductase releases minus the rate that free NADH binds to the reductase. That's the flux through this way. Then in this part, we can have the flux through this part of the loop, and that depends on the rate that NADH binds the oxidase minus the rate that the NADH bound to the oxidase unbinds. And then we can have also the flux through this part of the loop, and that depends on the rate that NADH is oxidized when it's bound to the oxidases to the reverse. Oh, oops, sorry. Shoot. Let me just. Okay. And this flux through this part of the pathway here is the flux through the electron transport chain. All right. Yes. Okay, and so this is ultimately something which we're going to be very interested in, is what is the flux through the electron transport chain in mitochondria. But I mean, one thing to note is that at steady state, all these fluxes must be equal. All right, and that by itself has huge implications. So essentially, because all these fluxes are equal, then you can put an equal sign there, like we have here, and that allows one to do some algebra, which shows the following. That basically in this coarse-grained model, you can relate FLIM measurements to metabolic fluxes through the electron transport chain. In particular, in this model, the flux through the electron transport chain is given by this equation right here, as I said before. And then because of setting all those fluxes equal to each other, you can write this as an effective oxidation rate for free NADH times the concentration of free NADH, where that effective oxidation rate is given by the following equation. It ends up that essentially all the factors here you can measure from FLIM. So you can measure the concentration of freely diffusing NADH from FLIM, the concentration of enzyme-bound NADH from FLIM, and this depends on also the difference between the ratio of those two and the equilibrium NADH bound ratio. And what we mean in this case by equilibrium bound ratio is what is that bound ratio when the flux through the pathway is 0. And if you assume in the absence of oxygen, the flux is 0, which is what we believe to be the case based on the known biochemistry, you can actually measure this beta equilibrium just by dropping oxygen. And so what this model suggests is that basically you can use FLIM measurements to measure flux through the electron transport chain. Well, the flux through the electron transport chain is actually also the oxygen consumption rate, if you assume that all the oxygen that oocytes are consuming is occurring in mitochondria, which it's a reasonable assumption, even though it's not, you could say a priori, it's not 100% clear whether it's true or not, but it's definitely a very reasonable assumption. Okay, so then we wanted to test if this model is correct. And so then what we can do is that we can say, well, we did this oxygen drop experiments. We have FLIM data as a function of oxygen. We can use that based on this model, or Jingbo can use that based on this model to predict OCR as a function of oxygen level. And then completely independently can measure oxygen consumption rate using biochemical techniques. And it ends up that the experimentally directly measured oxygen consumption rate quantitatively agrees with the prediction from FLIM. And so I think that's quite a strong evidence that this that there's something to this model. Okay, and so and again, there's no fitting parameters here. This is just just kind of direct inference from the model. Okay, so then we decided to basically use that model to try, and then we did other tests on the model, which I don't have time to talk about. But we tried to use that kind of framework to try to understand, well, what actually sets mitochondrial metabolic flux in oocytes, and that's the next part, which I'll tell you some about now. Okay, so like I said, so oocytes basically get all of their energy from oxidative phosphorylation. And so then what sets that rate of oxidative phosphorylation, which is equivalent to asking what sets the rate that oocytes consume oxygen. All right, so again, like I said, thinking about this kind of in a very coarse grain way, you could think about all of oocyte bioenergetics, in this diagram which I showed before, and you could imagine different scenarios about what could set the oxygen consumption rate. For example, a very reasonable hypothesis would be it's set by demand. So that's certainly true to some extent, at least at the organismal level, right? So you go on a treadmill, and then you start to breathe more heavily, right? And so because of the additional demand of you working out, you consume more oxygen. So demand is controlling oxygen consumption rate to some extent on the organismal level. Is that true on the level of individual oocytes? You could also imagine that maybe supply sets it. Like if you kind of force feed the oocyte, maybe it'll kind of run hotter. All right. Well, so one thing which is very interesting about this FLIM data is that not only does it allow you to predict oxygen consumption rate, like I said, in this model, you get that the oxygen consumption rate should be equal to the concentration of free NADH times the effective oxidation rate of free NADH. But you can actually independently measure each of those two parameters. So you can get the actual concentration of free NADH and its effective oxidation rate, in addition to the OCR itself, which is those two things times each other. Okay, so for a control oocyte, again, like I showed you, we get the oxygen consumption rate, and we measure these two guys separately as well. Okay, so then we can just start perturbing things and see what happens. So we're going to start by perturbing supply. All right, so for example, if you change pyruvate, like I said before, concentration, these things are largely consuming pyruvate, or you can do another metabolic perturbation, which has to do with supply, using oximate. You can use oximate, which perturbs lactase dehydrogenase, and you can see that when you do this perturbation, you can impact the concentration of free NADH by a lot, and you can impact the effective oxidation rate of that NADH by a lot. But if you look at those two things multiplied by each other, it stays basically the same. So in other words, perturbing supply impacts, like I just said, the effective oxidation rate and NADH and the concentration of NADH free, but does not affect either the predicted oxygen consumption rate or the actual measured oxygen consumption rate. What you can measure directly by, by biochemical means. Okay, so then what that shows is that perturbing supply has a metabolic impact on oocytes. It's not that it has no effect; it's actually strongly impacting metabolism, but it does not affect the oxygen consumption rate. Okay, what about demand? Okay, so Jingbo changed demand by doing many types of perturbations, including depolymerizing the spindle, depolymerizing, screwing up actin, screwing up protein synthesis, screwing up, ion gradients. And all those perturbations, again, have a significant impact on metabolism. They affect the concentration of free NADH in mitochondria and the rate that NADH is being oxidized, but they don't affect either the predicted or measured oxygen consumption rate. Okay, and furthermore, Jingbo has done a number of measurements, so some of this is still in process, but it seems like these perturbations also do affect ATP levels. So for example, when he depolymerizes a spindle, you actually get a significant increase in ATP levels in the oocyte. But again, you don't affect the oxygen consumption rate. Okay, so then what sets the oxygen consumption rate? It seems like it's not supply or demand. Okay, so what's another possibility? Well, it seems that, what about mitochondria themselves? So, if Jingbo it basically either, screws with mitochondria directly, either by increasing the amount of leak through, by adding a drug SCCP, or by screwing up ATP synthase, then that does affect both the predicted and the measured oxygen consumption rate. So in other words, this shows, I would argue, that the oxygen consumption rate isn't set in oocytes, isn't set by supply or demand, but rather by kind of what mitochondria themselves want to do. So they have just a certain rate they want to run at, and that's what's driving oxygen consumption rate. Okay, how? All right, this is still work in progress. One hypothesis is that oocytes, mitochondria and oocytes are the ATP synthesis is limited. So you could imagine the following scenario. So people for a long time have looked at purified mitochondria, and what they see is that if you look at oxygen in purified mitochondria, you look at oxygen consumption rate as a function of ADP level, you often see that they have a saturating function like the following. So this is data from, like I said, quite a while ago. So you could imagine that if mitochondria in vivo are behaving the same way, if the ADP level is in this kind of portion of the response of mitochondria, then you're in an ATP demand limited state. Like if you were to change ADP levels, you do change oxygen consumption rate. So that's kind of like a demand limited state. On the other hand, if you're in this part of the curve, if you change ADP levels by changing demand, you actually do not change the flux through mitochondria. In other words, they're basically saturated. We don't know if that's actually what's going on in oocytes, but Jingbo's in the process of trying to test that. Okay, so then in summary, we're using a combination of metabolic imaging with FLIM in a coarse grain model, and this allows measurements of metabolic fluxes and even enzyme kinetics in vivo. I say enzyme kinetics because you can infer this kind of effective oxidation rate of free NADH. It seems that supply and demand impacts the kinetics and concentrations of this kind of metabolism, of mitochondrial metabolism, but not the actual flux. However, leak in ATP synthesis activity does impact the flux. And so we're hoping that this is kind of a step towards a kind of a comprehensive coarse-grained predictive model for mitochondrial bioenergetics. Okay, and so then in the last few minutes, I'm going to switch gears and just briefly tell you about some other work which is related to this, which we have ongoing. So obviously, like what I've been talking about so far has been kind of like very fundamental questions about energy metabolism and mitochondrial function in oocytes. And another thing we're interested in is that can one actually apply these in a clinical setting? And so we have a lot of efforts in lab.
Yes. Before you move on, Jason Kahn has a question, which is if NADH is unavailable, what is the source of electrons for O2 reduction?
If you have if NADH is unavailable, if you don't have NADH, then you can't run well, I mean, so there's FADH as well, but if you don't have those two, then you can't run the like electron transport chain.
Okay, thank you.
Okay, and so now we're also in the process of, like I said, like asking if one could, you know, if these techniques could be clinically useful. And so the idea behind this, like I was kind of alluding to before, is that FLIM of NADH and also FAD provides a non-invasive way to measure mitochondrial metabolism. I told you before that there's a lot of clinical evidence of at least a correlation between mitochondrial metabolism and the viability of oocytes and associated embryo. And hence that suggests that maybe instead of using just morphology to select the best eggs in embryo, one could use FLIM. All right, so that's the thing which we're interested in exploring. And so to do that, we're collaborating. We have a number of collaborations with different IVF clinics, and we're doing this in many different ways. So one thing is we're getting immature human oocytes. So these are actually clinically useless because people don't know how to properly mature them. And so when I say immature, I mean that basically like oocytes, when you collect them, they're supposed to be in metaphase 2 and having kind of a spindle there, waiting to be fertilized. Some oocytes they get from patients are in an earlier stage. It ends up you can mature them in vitro and get them to have what looks like a perfectly good spindle, but there's clearly something wrong with them, because even though, as far as, just kind of morphologically, everything looks fine, when people fertilize those immature human oocytes, they actually only produce healthy offspring at a very, very, very low rate. And so they're not used clinically. And so they're discarded. And this is work led by Marta, a grad student in the lab. A lot of the work I'm about to mention right now. And so she's been studying the mitochondrial, she's looking at mitochondrial metabolism in these in vitro matured human oocytes. In addition, oocytes are metabolically coupled to surrounding cells called cumulus cells. Those are also retrieved clinically when oocytes are retrieved. And so Marta has been looking at cumulus cells from actual IVF patients. And so normally those will be retrieved and discarded. And so then she's been doing metabolic measurements on them to try to understand how metabolic measurements of cumulus cells are associated with stuff like age of the patient, whether the corresponding embryo actually successfully produced a child, etc. And then also, we've been doing a lot of work looking at using the same techniques to look at embryo development. And so what you're seeing here is a 5-day continual time-lapse movie of mouse embryos developing from one cell stage up to what's called the blastocyst stage, which is that they would then hatch out of the surrounding eggshell, what's called the zona pellucida, and then implant in the mom. And trying to understand also how these metabolic processes play out in the developing embryo. And we definitely see some very interesting things in that regard. And I also just think it's cool that you can take movies for 5 days and watch all this stuff in detail. And then we placed a microscope in a FLIM microscope in a IVF clinic at Boston IVF. And Marta has taken a lot of data on discarded human embryos. And so these are embryos which clinicians decided mostly that they thought they wouldn't be viable. Yeah, so these are mostly ones which clinicians decided probably wouldn't produce a healthy offspring, or some of them are embryos which the patients no longer desired. And so then we have done a lot of FLIM measurements on those, and we're looking at trying to understand FLIM parameters of those guys and metabolism of those guys, including for many of those we actually do have by biopsies we actually know the state of chromosome segregation errors, and so trying to look at least correlations between chromosome segregation errors and metabolic parameters. as well as using this to test whether this technique can safely be used on embryos, which would actually be implanted back into a person. So in conclusion, so we're investigating many different aspects of energy metabolism. We're looking at energy, production, if you will, like it's really transduction. But in other words, mitochondrial metabolism, trying to look at kind of energy consumption, trying to understand what is the energetics associated with spindle assembly or signaling in the spindle or other functions of oocytes. And also trying to understand the impact of varying energetic fluxes. And so like if mitochondria are screwed up, how does that affect spindle self-organization behaviors, chromosome segregation, errors, etc. And as I said before, looking to see if potential clinical applications of this, of FLIM. And I'll just mention that this line of work, like everything I'm telling you about here has been really based on FLIM, and maybe I mentioned a little bit of stuff having to do with metabolic sensors, fluorescent metabolic sensors, but I've gotten, this type of work has gotten me very interested in these kind of questions of cellular energetics more broadly. And we're starting to do a lot of other types of work in this general area. And so with that, I'll just say that, most of the work I talked about was Jingbo's work on kind of, this kind of theory and quantitative measurements of mitochondrial metabolism. Also the, work on human eggs and oocytes and embryos and cumulus cells. It was Marta, and then Tim Sanchez also did some work related to this as well. Thank you.
Cool, thanks a lot. I really appreciate that. So I've missed a few questions along the way. So Steph Weber wanted to come back to a question which I wanted to, and another person did too, which was, she says, what is the hypothesized connection between mitochondrial function and spindle assembly stability, ATP availability, or something deeper? So I think that, maybe the point is that as you transition to the final part, you didn't circle back to that point, or at least if you did, it went over my head.
No. Yeah, I mean, this is all very, very much a work in progress. And so I didn't, I didn't touch on that. No, I mean, I think actually answering the main question about the connection, that's a very, I mean, again, we're trying many different things, so it's a quite hard problem to say something definitive. So, you could imagine, and people have imagined all sorts of different scenarios, I mean, one possibility is literally ATP production. Another popular hypothesis is this related to reactive oxygen species being formed by mitochondria. You could certainly imagine it's also involved, it's rather about ion homeostasis or other possibilities. And so, yeah, there's a huge number of possibilities.
Okay. A technical point from Scott. Is it a concern that FLIM or other fluorescence imaging could cause some amount of DNA damage in embryos?
Definitely for clinical applications, that's really a concern. And so that's why, and so we've done a bunch of safety studies in mouse eggs and embryos, including, doing measurements on like extensive FLIM measurements on developing embryos, like I showed some of, and then, transplanting them back into mice and then seeing, do they give rise to like pups that seem healthy? Yeah. Even stuff like that, it's not definitive. And we're trying to do more safety studies on discarded human embryos. It's very hard to show definitively that you haven't damaged stuff, though.
Mm-hmm. Mm-hmm. Rudolf Oldenburg had a question for you. First of all, he says, hi, Dan. So that's good. Regarding spindle integrity, I seem to remember that mitochondrial metabolism generates free radicals in the cytosol. Therefore, mitochondrial metabolism is reduced during mitosis, starting with nuclear envelope breakdown for reducing the damage to genetic material now exposed to the cytosol and its free radicals. Might there be a link between spindle integrity and the functioning of this downregulation of mitochondrial metabolism? So, I think it's actually in the same spirit as what Scott asked. Yeah.
Yeah. So like I was saying before, so definitely the idea of, and so for example, there are many different hypotheses on what causes age-related infertility. And so I'll back up even more broadly. So like I said before, so we know that kind of like it's definitely chromosome segregation errors is definitely a problem, and then many different people have proposed many different hypotheses. For example, like in kind of the more cell bio community, one idea which is very, very popular is this: it's about the connection between chromosomes breaking down, and so the cohesion between chromosomes. And that could be correct as well, and it could be connected. And so, for example, it could be the case that if you have misfunctioning mitochondria, you're more likely to have free radicals being generated, which are more likely to damage proteins, including those which are responsible for kind of holding chromosomes together. And so these things could be connected or not. I would say, like, we really just don't know at this point.
Gotcha. Maybe what? Go ahead, Ernan. Sorry.
I was going to ask the question Han-Jen is asking, which is, can calorimetry also be used to distinguish screwed up mitochondria from normal ones?
Yeah, that's a fantastic question. And so like we've become very, very interested in doing calorimetry, and so we actually have a lot of work, unpublished work. Hopefully that's being published on using metabolism, or using calorimetry to look at related things. Yeah, that's a fantastic question, and I think it's a really interesting way to probe that. Yeah.
Ricky Garner wanted to hear your thoughts on how these results relate to what Janne talked about yesterday, which was the known scaling of mitochondrial volume with cell volume. Any thoughts about that?
Yeah, and so I mean, so like mitochondria and oocytes are kind of very special in the sense that, well, like one, they actually even morphologically they're quite different from kind of the normal mitochondria that we're used to from textbooks. And so they're often considered to be immature mitochondria, and also they don't replicate at doing the stages which I was talking about. And so basically, like, yeah, and so there's not a lot of the really cool cell bio either involved in mitochondrial replication or fission and fusion going on and at these stages in either the oocyte or the early embryo. And, having said that, there also is, people argue that there's a huge amount of variation in number of mitochondria between oocytes and in both human and mouse. But I think that's not, yeah, but that's not well understood either. So here I think it's quite different from other.
Okay.
Thomas, did you want to add something?
Yes. Following on that, can you test the hypothesis that long, big mitochondria have a different flux than small roundish ones, or relate? Because there have been contradicting data on that in the literature, and you seem to have a more direct measurement.
So I think that's a super interesting question. And so like, so then if you believe our model, which I do, and you should believe it as well, then you could take, FLIM measurements to tell you what flux is in mitochondria, and that you can do on subcellular resolution. And so like, we also have some very interesting preliminary data showing that there's at least spatial patterns. So like here, everything I was saying, like we kind of treated all the mitochondria in the oocyte as if they were the same. Actually, even just with the FLIM measurements, if you break stuff down, you can see that there's differences in mitochondria depending where they are, and doing stuff on the morphology would be incredibly interesting as well. And yes, I think that would be feasible.
Yeah, one question that we have is, this is, I think, related to the IVF techniques, is could you culture the oocytes at low oxygen and just pick the ones that survive?
And so yeah, no, that's definitely quite interesting. I mean, like, of course, like human oocytes and embryos are very delicate, and people tend to try their best to perturb them as little as possible. And so they try to do stuff in what they think is like the optimal conditions, even though there's some debate about exactly what those are. But I think it's definitely an interesting question. So like I said, so like it's unclear if there's a causative relationship between mitochondrial metabolism and there's definitely a correlative relation between mitochondrial metabolism and oocyte health and embryo health. if that's causative, it would be really interesting, kind of building off of that, to ask, well, then can you adjust mitochondrial metabolism to make it kind of good, whether by manipulating it with, like oxygen or nutrients or drugs or whatever. I think that's obviously a super interesting direction.
And I guess so. Is your claim that if you did that, like when, like you fix the embryo at that early stage, like would that be like how heritable is this fix, and how heritable is the damage of the mitochondria? I'm curious.
Well, I mean, like I think at least most of the changes that we're talking about clinically are related to age-related infertility, and so then in that sense, I mean, like maybe the onset of age-related, maybe the age at which you start having the onset of age-related infertility, that could be heritable, but like the fact that like age-related infertility, it's something that's like an environmental thing, right? Because it's like you get screwed up with age, right? You know what I mean?
Yeah. Okay, fair enough.
I guess Thomas gonna ask something, then I will also. Go for it, Thomas.
Yeah, so my other question, it relates to, Pat O'Farrell did some really nice work that showed that, at least in flies, there is a bottleneck. So, mitochondria are hugely polymorphic in terms of their own mitochondrial DNA. And so when you look at the cell, in fact, you have a collection of different mitochondria which have different, genetically different, genetically defined mitochondria. And so question one is, do you have a bottleneck in the formation of an egg in Xenobus like it is in Drosophila? Like amongst the many mitochondria, you select a subset of those that will be in the egg and then will grow. So, and second, could you relate, in other words, the status of activity of mitochondria with their genetic polymorphism by sort of comparing, in your assay, FLIM data and, I don't know, sequencing of maybe this is a bit difficult, but I'm trying to relate these two different subfields, which I think is really interesting.
Yeah, and so
to assess fitness in a way.
Yes, yeah, okay. So then to both those points, okay. So one in mouse and human, which is what we're doing these experiments on. There also is a bottleneck, and so that's definitely been established as well. Another thing is that so our work, for example, with cumulus cells, and this will be the case in human oocytes, even though we haven't done it yet. But with cumulus cells, we collaborate with Dagan Wells, who's an expert at the relevant genetic techniques. And so we've been doing work sequencing the mitochondrial DNA to ask about polymorphism, both asking. So for example, so like we have this the cumulus cells that we've gotten are from actual patients who've actually gone on are outside people out there in the world, and so we both asked, to what extent is there correlations between FLIM parameters and genetic variants in mitochondria that are segregating, and so far we haven't seen any. And also, you can also get a sense that heteroplasmy from sequencing, which like you were talking about, so within an individual, and also so far we haven't seen any correlation between those and the FLIM measurements.
So Dan, I think I'll maybe ask the last question, which is from tomorrow's speaker, Lena Kostlover. I like her question. So she says, following up on that, but I don't know what that is, but I'll just ask the question because it makes sense. Are there regions of the egg that reliably have higher mitochondrial ATP production flux? For instance, is there more ATP production locally where the spindle is being formed than in other regions of the cell? So, the general question of heterogeneity spatially and temporally, is damn interesting. I'd love to know more about your thoughts on this. And as you probably remember, in my group, thinking about doing, well, they've been making some headway on that kind of stuff. Anyway, so I think that's going to be the last question.
Yeah, and so yeah, that's a fantastic question. And following up on that, we do have indication of something interesting happening around the spindle as far as stuff, but this is still early days, and we don't know what it is. And if you do see differences, of course, one has to again disentangle cause and effect. Like, is it that
Yep.
the mitochondria which are different, which localize differently at different places, or are the mitochondria the same everywhere, and then they're responding to different, like kind of, gradients in other things. And so there's definitely interesting going around and happening around the spindle, but yeah, we don't understand it yet. Definitely, that's the thing I'm very, very excited about.
Yeah. Okay. Well, so thanks a lot to everyone out there. Tomorrow we have Lena Koslover from UCSD, who's going to talk to us, I think, about transport in ER, if I remember correctly, but I might have that wrong. But in general, whatever she decides to talk about is fun. And so with that, I wish you the best for today, whatever that means. For Toma, I know that means good evening, but for me, it's still the morning. And go out and have a great day. Be healthy and safe. And thanks for tuning in.
Bye, Dan. Thank you so much.
Ciao.
Bye, everybody.